The ELISA assay, enzyme-linked immunosorbent assay. I always feel silly calling it the ELISA assay because the A in ELISA stands for assay. So it's almost like when you say ELISA assay, it's like saying the ATM machine. The M in ATM stands for machine. So it's the automatic teller machine. So this is the enzyme-linked immunosorbent assay. Um, This is based on the RIA, the radioimmunoassay, first described in 1960. Developing ELISAs began in 1966 to 69 when antigens and antibodies were coupled using enzymes. During the 1960s, enzyme linking procedures were developed by two separate groups under Avramius and Pierce, and immunosorbent technique was developed by Wyde and Jerker-Porath. The technique for ELISA was published in 1971 by two independent researchers, and this technique has been around ever since then. What the ELISA assay consists of is a couple of different steps. So first of all, what we've got is we've got to use a polystyrene plate and affix to that or coat to that an antigen or an antibody. Whenever that antigen or antibody is affixed to that plate, you can remove the excess solution from that plate. Then what you're going to do is a blocking technique, very similar to what we did with the Western blot. So we'll use a solution of BSA or we'll use milk or something to block it. Then we're going to remove that, and we are going to go on to add the antibody for the antigen that we previously added to our plate. And so if that antigen, if the antigen that we are looking for is present, that antibody will bind to it. That antibody has a label on it, or it has, well, an enzyme. Hence the E of ELISA. What we'll do to that is we will add this enzyme substrate. And so whenever we're doing that, what we're going to be doing is we're going to add something that actually produces a small amount of light. And based on whether or not that antibody is present and binds to the antigen that we were kind of probing for, we will see some sort of enzyme activity. Okay, so one way that you could kind of look at this is, or another way that you could look at this is this black line down here. That's our polystyrene plate. To that we have an antibody specific to our target antigen permanently affixed to that surface. Okay. Then what we'll do is, let's say a blood sample. We will take that blood sample, and actually the first thing that we'd take that blood sample, load it onto our ELISA plate. We'll incubate that for a little bit, and we will have a protein that we are looking for that is going to be bound by this antibody that we have affixed to this polystyrene plate. What we'll do is, after a set amount of time, we'll wash off that solution, get rid of all the excess solution, and discard it. Our protein that we were probing for, well, that protein is interacting with our with our antibody that is on this polystyrene plate. And then what we'll do is we'll add our second antibody. The second antibody also binds to our target protein of interest, but that second antibody has an enzyme. And then after that incubation has taken place, we'll add the substrate for that enzyme. And the substrate for that enzyme will be producing our product, or will be used to produce our product. Okay, so now let's go back to the starting point. If our protein is absent from this blood sample, well, then this isn't going to happen. And if that doesn't happen, well, will this second antibody bind? No, it won't. So we won't have that actual activity there. Okay, now there are a couple of different assays, or a couple different derivatives of the ELISA assay. ELISAs are commonly used in the healthcare system, justice system, and food industry every day. A direct ELISA is the antigen is immobilized and you use one antibody. What we just looked at was known as an indirect ELISA, or sorry, that was a sandwich ELISA, and that sandwich ELISA has an antibody which binds to a protein, and that protein binds to an antibody. An indirect ELISA would be something where you have two antibodies that are present, and you're able to actually deduce what the antibody concentration is. Now, I would say the most common form of an ELISA assay is this direct ELISA, in which case, you would have your plates with antigens basically affixed to them, immobilized on there, and then you can just run those antibodies over it and say whether or not that's actually present. All right. Now the benefits of ELISA: one, they're quick, efficient, and inexpensive. They're very, very sensitive. So because you have a enzyme that catalyzes a reaction, you can standardize it against internal controls. The color spectrum can be evaluated with a spectrophotometer, or you can determine the absorbance of a sample. You can run several different reagents at the same time. The absorbance is used to evaluate and quantify concentrations from a standard curve. So you have a quantitative measure here with an ELISA assay. So an ELISA can be treated as a quantitative or a qualitative assay. Quantitative assay gives you the opportunity to actually sort of standardize it, and you're measuring the optical density or fluorescent units of the sample. So you're using the enzyme, you're standardizing it against, well, how many products are actually being generated. A qualitative measure, you can use an ELISA assay for a simple positive or negative. However, if you really kind of comb into those or look into those details or look into that data very specifically, what you'll find is that if there's a very, very small amount, well, then you're not actually going to get a positive result. And a very, very small amount of your antigen, well, then you're not going to get a positive result. And so you do have kind of extreme ends there that you can land at. Okay, now how is this technique done? The basic procedure consists of a couple of different steps: coding, blocking, detection, and readout. The coding is the first layer of your antigen is absorbed to the surface of the wells of your 96 well plate. Or, if you were only doing one assay, well, your plate. But you need to apply those antigens or your antibody to the surface of that plate. Then you're going to do that blocking using either BSA or milk or something else. And then you're going to detect whether or not your protein is present. And then finally, you're going to get your readout, where your substrate is catalyzed by the enzyme bound to the detection agent and generates a colored readout. Between each one of these steps, you're going to rinse that plate, or rinse your adherent surface. There are many different types of ELISA assay, which we've already kind of talked about those. But indirect and direct are your most common procedures. Now, the direct detection method: a sample containing the antigen is prepared and incubated to allow the antigens to adhere to the walls of the plate. So here we've got down here, we're basically setting the stage for that green ball to adhere to the surface. Then we're going to use BSA or milk to prevent non-specific binding in wells, and the plate is washed again. Okay, so we'll add milk or a BSA solution. I lost my cursor. BSA solution to that right there. Antigen-specific antibodies are added after that blocking, and those are going to bind to your antigens. Excess antibodies are removed by a third wash, leaving only the active antigen antibody complex in the well. So this is our antibodies that have specifically found our antigen of interest. The antibodies have an enzyme affixed to them, hence that blue ball right there. And those enzymes are able to catalyze a reaction once our substrate is added. And this substrate, let's say our product is going to be this kind of yellowish amber solution. So that is an indication that our product is actually present. Quite commonly, the enzymes that are used are phosphatases, so alkaline phosphatase, or HRP, which is horseradish peroxidase. The enzyme reaction can be measured and quantified using the spectrophotometer, and the antigen presence is demonstrated by the colored or fluorescent signal for the enzyme reaction. So basically, if you can treat that, like I said, as a positive or a negative. You can say, oh, well, my solution turned yellow, so my antigen is present because my enzyme is present. But you could also use that spectrophotometer to say, here's how yellow that solution got, in which case you can correlate that to the amount of your enzyme. The indirect detection method is similar to the direct, but what we've got is we go through that same process where we're adhering our antigen to the surface of that plate. Then we have an antibody that binds to that antigen. This is known as your primary antibody, similar to what we talked about previously. Then we use a secondary antibody. Whenever I said previously, I mean with respect to Western blotting. Now, this secondary antibody has the enzyme bound to it, because that's the one that's going to be a little bit more general, and it's going to detect your primary antibody. Now, when that second antibody is present, that tells you that the primary antibody is present. That primary antibody tells you that your antigen is present. Now, if you did not have that antigen present, then your primary antibody would have nothing to bind to. And if you don't have anything for your primary antibody to bind to, well, then you don't have anything for your secondary antibody to bind to. Again, this is a fairly quantitative technique, and you can see this color change as an indicator of enzyme activity and present, therefore, or indirectly, presence of your antigen. Okay. Now, comparisons of these two techniques: direct. The direct is very quick. You do not have to worry about a secondary antibody detecting something else or anything like that. A disadvantage is cost. There is a cost with developing that primary antibody that has that enzyme linking, so that's going to be fairly expensive, but it also can have a negative impact on that antibody. So labeling primary antibodies is time consuming, and labeling primary antibodies could potentially damage those primary antibodies. There's no flexibility in choice of primary antibody label between experiments. So you've developed an antigen, or you've developed a primary antibody for one specific antigen, and it's very effective against one specific antigen, and that's about it. You don't have to worry about two primary antibodies or two secondary antibodies binding to that primary antibody. An indirect ELISA is, well, since you're using a secondary antibody, it's an extra expense, but that secondary antibody, you have some reliability that it's functional and that it works well, even with that enzyme bound to it, so you don't have to worry too much about that. That secondary antibody can be used that secondary antibody can be used with a number of different primary antibodies, so you don't have to worry about developing a new one of those or anything like that. Disadvantages are, well, you're introducing another antibody, and so that could potentially negatively impact your results and, change your results to some degree. It does also require some more steps, so there's a time cost when actually doing that experiment. Here's all the sources for that.